Thank you all, obviously, for supporting medical research at WRI. This is a fantastic place and only growing. I promised Andrew and John and pretty much everyone else who works here that I won't put on a medical school lecture, but I am going to share with you some scientific details and some background on the cardiovascular program we're trying to grow here. I promise I'll keep it pretty accessible, patient-focused, and hopefully engaging for everybody. So as John said, I'm a cardiologist and a cardiovascular researcher. I spent part of my life caring for people with advanced heart diseases, and the other part of my life trying to figure out why heart diseases develop, and trying to figure out how we can leverage new technologies to help the patients of tomorrow. The work I'll show you tonight sits at the intersection of patient care, spatial medicine, and artificial intelligence. But at its core, it's driven by a few basic questions. Can we find disease sooner? Can we understand it more deeply? And can we use that knowledge to help patients live better lives? Over the next 15 minutes, I'm going to walk you through some examples of how technology are helping us to improve the lives of cardiovascular patients and generate new discoveries, and how these new discoveries are helping us improve diagnoses, achieve better risk predictions, and ultimately achieve more personalized care for heart patients. So as John said, cardiovascular disease is the leading cause of death worldwide, unfortunately. More than 18 million people die from it each year, and more than 500 million people are living with heart disease today. Here in Australia, every 80 seconds, someone is hospitalized for heart disease, and around 4 Australians will die from a heart disease-related complication. Despite enormous progress in treatment, cardiovascular medicine still faces central challenges, one of the biggest of which is that we tend to find disease too late. Coronary artery disease is typically found only after dangerous blockages occur, and heart muscle damage typically is found only after years of scarring result in heart failure. And in the field of heart transplantation, which is near and dear to John and I, we still rely on regularly sampling pieces of our patients' new donor hearts to see just how much rejection and damage is currently going on in there. In other words, we're really quite good at identifying yesterday's injuries, but we're still not nearly good enough at identifying tomorrow's risks. In order to make the next generation of breakthroughs in cardiovascular medicine, that needs to change. Simply put, cardiovascular disease almost never starts when a patient starts feeling unwell. Biology happens first, then symptoms develop, often much later. Blood vessels start narrowing, heart muscle cells start weakening, an inflammatory reaction develops, scar tissue begins to form. These changes are typically invisible to patients early on. And more often than not, they're invisible on my standard battery of clinical tests early on as well. Gradually, tissue injury accumulates, and only then do we see what we recognize as disease: chest pain, breathlessness, swelling, symptoms of heart failure, arrhythmias, and so on. Unfortunately, when we detect disease only after significant damage has occurred, it really limits our options as doctors trying to treat it. We might be able to treat you, but we can no longer prevent the problem from happening, or we might be able to slow progression, but it may be too late to restore some of the function that's been lost. The future of cardiovascular medicine needs to move towards earlier detection, better predictions, and ultimately more personalized solutions for heart disease. The rest of this talk is really focused on that core idea, going closer to the source of disease, using the newest technologies to probe disease biology hidden there, and building research programs focused on turning these discoveries into breakthroughs for patients. So what does it mean to go closer to the source of disease? For us, it means studying human cardiovascular disease in human heart tissues. For decades, researchers have relied heavily on simplified systems to study heart disease. These are cells in a petri dish, or experimental models, or animal models of all kinds. Those systems are important. They allow you to study mechanisms in isolation and test out new ideas. But human heart disease doesn't happen in isolation. It's not a controlled experiment. It's shaped by the complexities of real life: aging, lifestyle, diet, environment, genetics, medications, years of change, adaptation, and injury. Human heart tissue gives us a way to study all of that real-world complexity directly. It lets us examine disease in its native environment, and it gives us access to the biology that actually matters to patients. Once we have access to that human tissue, the next challenge is getting as much information out of it as possible. That's where digital pathology and artificial intelligence become powerful allies for us. The first step is straightforward. We take traditional glass microscope slides, and we turn it into high-resolution digital images. Once the slide is digitized, it becomes data like everything else we digitize. AI-assisted digital pathology allows us to analyze that data systematically, detecting subtle patterns human eyes may not see, and converting our visual observations into concrete quantifiable measurements. So instead of simply describing tissue architecture qualitatively, as physicians and as scientists have done for generations, we are now measuring it quantitatively. We're measuring blood vessel structures, and we're measuring scar burden, and we're measuring the density of inflammatory cells and many other features. And we're quantifying these reproducibly and at scale across hundreds of thousands of cells. This allows us to ask whether these tissue patterns are linked to clinical questions we care about, like a diagnosis, treatment response, outcomes, and future risk. In other words, digital pathology and AI tools help us turn tissue structure into measurable information we can connect back to patients. But tissue structure is really only half of this story, as we sort of already alluded to. The other half, and the deeper layer, is this concept of spatial biology. Just as AI digital pathology allows us to learn the nuances of tissue architecture, advanced spatial biology methods help us understand what the cells in that tissue are actually doing. With spatial biology, we're not just quantifying tissue structures, we're actually measuring hundreds of biological signals from within those structures. That means we can ask deeper biological questions. Which types of immune cells are present? Which types are clustering around blood vessels versus which types are clustering around heart muscle cells? And are those heart muscle cells showing signs of stress? Are they showing signs of metabolic dysfunction? Are they showing signs of stiffening? This is where human tissue becomes a biological map of disease, an atlas of cells and signals and neighborhoods and injury patterns. Within that framework, I think we're going to kind of probe into a few examples here that really highlight what I mean by all this and what John meant by all this, especially in the context of cardiovascular disease. We'll start with an example from coronary artery disease, one of the most familiar and most common heart diseases, but one where our new methods are really helping us make new discoveries for patients. So most people think of coronary disease as a plumbing problem. Cholesterol builds up, arteries begin to narrow, and then eventually patients may experience chest pain or a heart attack. That view's not wrong, obviously, it's true, but it's incomplete. Most of what we know about coronary disease in human hearts is learned from studying hearts at the very end of the disease process. These are patients with advanced heart failure and extensive scarring after many years of accumulated damage. The problem is at that stage, you're really only learning about what a severely damaged heart looks like. You actually learn very little about the biology that initially drove that disease forward. And that's the missing link here. What we wanted to know is whether we could learn about what was happening in coronary disease earlier, before advanced heart disease had set in. To answer that, we'd studied a unique cohort of human heart tissues from patients who have coronary disease but have no evidence of advanced heart failure or decreased function of their heart. And by combining large-scale proteomics with AI digital pathology tools, the ones we just talked about, we found that early stages of coronary disease are far more than just cholesterol buildup in arteries. We found that it also produces coordinated early changes in the heart muscle itself. These are changes in the small blood vessels, changes in the energy-producing machinery of heart muscle cells, and changes in the supporting tissue between heart cells. This stuff is important because it's these early, silent, hidden processes that we need to interrupt if we want to stop coronary disease from becoming irreversible damage tomorrow. And with an eye towards the future and related to what John said, it's these hidden early disease mechanisms that we're really going to be wanting to target if we want to make the next generation of breakthroughs for coronary disease patients. I think more broadly, the core concept here, finding hidden biology which underlies future injuries, really motivates a lot of the projects we do here at QSBC, both cardiovascular and cancer. And it's particularly actually important in the next example I'm going to show you here. So, heart transplantation. It is a miracle of modern medicine. A donated organ is an amazing gift. It can promise years or even decades of high quality life for the people who receive them. But after a transplant, patients live with a constant balancing act. They need enough immunosuppression medication to prevent them from hurting their new heart. But they need to have not too much immunosuppression or else they risk jeopardizing the rest of their body through infections, through kidney injuries, and through cancer risk. In current clinical practice, post-transplant medicine is a largely one-size-fits-all approach. Patients undergo frequent protocol-driven biopsies, clinic visits, blood testing, all on a pretty scheduled basis, with deviations from that schedule really only occurring after something bad has happened to them. In other words, we practice very reactive medicine in transplant, really informed by what's already happened to people, instead of proactive medicine based on a deep understanding and accurate forecasting of their future personal risk. In an effort to change that paradigm and help patients do better, we recently asked the following question. Can we use the data we already collect from our patients to predict their future risk of rejection? We combined AI digital pathology from their biopsies and routine clinical data from all of their visits, and using AI modeling methods, we combined these to assign patients to different risk tiers for future rejection. The results of this effort were striking. Patients identified as high risk had 6 times the risk of rejection of patients with normal risk, and patients deemed to be low risk had half the risk of rejection of normal risk patients. When you take that information together, you begin to see a door opening towards ways we might be able to personalize people's care and management based on their personal and individual risk profiles. So maybe for a low-risk patient, that will mean having fewer clinic visits, fewer biopsies, and lower doses of immunosuppression. Whereas for a higher-risk patient, it might mean increased surveillance, more testing, and targeting a slightly higher dose of immunosuppression to see if we can't avoid a bad event happening next month. We really believe this is the start of a progression in transplant medicine from protocol-driven care to risk-adaptive personalized care, and we're really excited about the direction of projects like this to deliver that kind of care for our patients. Our third story tonight introduces a slightly different heart disease challenge. Sarcoidosis is an autoimmune disease where the immune cells attack the body and form these unusual cell clusters called granulomas. When this happens in the heart, patients can experience rhythm disturbances, heart failure, and actually have a very high incidence of sudden cardiac death. In fact, most cases of cardiac sarcoidosis are diagnosed at autopsy. A major challenge in cardiac sarcoidosis is establishing the diagnosis in the first place. The problem is that these granulomas that define the disease are rare and they're scattered randomly throughout heart tissue. So if I were to refer a patient for a biopsy because I have some suspicion that they may have this, they'll get their biopsy, but more often than not, that biopsy comes back with no granulomas, which means we do not make the diagnosis, which means we do not initiate timing therapy, which means we do not prevent bad things from happening to them. We wanted to see if the use of new technologies could help improve our ability to make this key diagnosis. Using advanced spatial biology methods, we found that sarcoidosis affects tissue far beyond those characteristic visible granulomas that define the disease. Areas that looked normal under a conventional microscope contain dozens of abnormal immune activation and tissue remodeling signatures. In fact, when we used these hidden tissue signatures, we were able to correctly diagnose 96% of the tissues that conventional microscopy failed to diagnose. We are now working at WRI to try and translate this initial discovery to something that might actually be clinically deployable as a tool using the spatial biology instruments we have here, with the goal of making more accurate diagnoses, initiating more timely therapy, and ultimately helping patients do better. This story is, I think, a really great example of why we're so excited about spatial biology, right? It's not just a means to make beautiful PowerPoint slides, although it is good for that. They are fetcher, but it really gives us a way to see things in the tissue that we previously couldn't see. And things that aren't just academically interesting, but things that might be able to touch patients' lives. The 3 examples I think we covered here are 3 very different disease processes, but I think there is a common thread linking them all together. Clinically meaningful biology is often present before or beyond what our conventional tools can find. In coronary disease, we learned that tissue injuries are present long before symptoms develop. In transplantation, we found tissue signatures that don't just tell you how a patient's doing today, but it's going to tell you how they're doing next week and next month. And in sarcoidosis, spatial biology unlocked key features of the tissue that allow us to make crucial diagnoses where conventional methods would have failed. Taken together, these examples highlight a larger opportunity here, pairing spatial biology with clinical expertise to turn the latest technologies into real personalized medicine breakthroughs for patients. We're building a cardiovascular program which we hope will seize that opportunity. Our program starts with clinical expertise in the diseases we're actually interested in researching. It focuses on studying these diseases in their native environment in human tissues, and it uses advanced technologies to uncover biology that conventional methods miss. At the center of this program is a simple idea that better patient care begins with better disease understanding. And to make this a reality, we're investing and we're continuing to invest in the key resources that made all of this discovery work possible. It's the people and the instruments, the tissue resources, the computational tools, and the key strategic partnerships. Because these are the key ingredients for building a world-class research program. And that is exactly our long-term goal here. We aren't just trying to do one excellent science project. We're trying to build a hub, center here in Brisbane, but with connections around the country and around the world for high-impact cardiovascular research. Given that vision, one of the essential foundational steps we're trying to take in the short term is consistent access to the human tissue that feeds our ideas. At the moment, we still rely heavily on international collaborations and partnerships to get the tissue we need to run our experiments. Those partnerships are, of course, incredibly valuable, and they're part of building a world-class program. But one of our goals is to expand that capability right here in Queensland. The pieces for this foundation infrastructure are already here in the city. We have biobanking capability at WRI. We have laboratory and surgical expertise at John Fraser's CCRG and up at the Prince Charles Hospital. We have spatial biology expertise at QSBC and at our academic partnerships. And we're trying to build now is a way to bring all of these together to create something known as the Australian Human Heart Tissue Bank. And why are we doing this? We're doing this because we don't just want to run one experiment. We want to build enduring capability here in Australia to make the next breakthrough in cardiovascular care. The reality is philanthropy is important in these types of discoveries. It's because the capabilities and goals we talked about today aren't just going to form overnight. They require the people and the platforms, the partnerships and the pilot discoveries. They require the freedom to pursue ambitious ideas before traditional funding mechanisms will support them. Philanthropy helps build all of those bridges. They help bridge these great ideas and allow us to achieve patient impact. It supports the people who do the work and the platforms they work on. And it supports the early momentum that allows a discovery to become a major fundable program in the future. At places like WRI, it is not an exaggeration to say that philanthropy is really the foundation of most of our discoveries, and we're grateful for all of you who've contributed to it. With that, I'm going to wrap things up. I thank you all for your attention. I'm happy to take some questions during the next session.
Thanks very much, Eliot. A couple of things stand out to me. One, how much we don't know about the human body and about how much we're just learning with these new tools, techniques. And a couple of things that John Fraser bangs on to me all about often, how important the patient is at the centre of the research, making sure we're very clear about the impact and why we're here. I think John uses an analogy. It's like having a race car without wheels if we're not involving our patients, and how important they are. And the second thing he continuously, usually with a football analogy, bangs on about is how much of a team sport the research is. And Eliot touched on that. The collaborations we're building and people like John, Arutha and Eliot are really helping us. Public sector, private sector, Prince Charles is the biggest cardiac hospital in the country. St Andrews and the Wesley here, but also academic partners with Institute of Molecular Bioscience at UQ and the Fraser Institute. And we're all working together. And then we throw in the University of Pennsylvania that Eliot's brought to the table as well. And there's a number of other universities around the world that are trying to crack these massive problems. And it's just such a thrill to see. And I think that's one of the things that Brisbane does have going across some of the other bigger centres down south. We really play nicely in the sense that we want to collaborate with people. We're not in competition. We're all working together to crack some of these key healthcare problems. But now we're going to change pace. Ruth is going to come up and lead the discussion with our panel. So just a little bit of background on Arutha. Associate Professor Arutha Kilsinger is the world's first chair in spatial medicine. He's one of the founders with John, and he's the scientific director of our Queensland Spatial Biology Centre here at Wesley Research Institute. Arutha also leads the clinical omics lab at the Fraser Institute of UQ, and he's pioneered spatial transcriptomics using digital spatial profiling approaches in the Asia-Pacific region, contributing to world-first studies for lung cancer, head and neck cancer, and COVID. Please welcome Arutha to the stage and this will lead us through a discussion with John and Eliot.
It's an absolute pleasure to be here, and I was just reflecting as I was coming in 12 months ago, it was so different. And then where, where the research has come to and how it's evolved. We've got a number of the team here, and we spoke about the team. Could some of the teams please stand up that support this research? My team. And these are the people that keep the labs, operational, and they're critical to all the research that we get to talk about. There's probably another 20 much people around different places in Brisbane today. So thank you for supporting all of that. So John, maybe we'll kick things off with you. spatial biology, you've seen this evolution play out in cancer, and what are your thoughts around how this is going to be impactful for cardiovascular medicine?
Thanks. But I think the patient I talked about this for, Eliot really said, once we see patients in intensive care, it's truly, this man said with you've also an ulcer, and then there's next for second. If you done it, you'd be dead. And if we can get into this earlier, not just give people more, add years to their life, but add life to their years. And as you says, Eliot said nicely, the sarcoid story, but as I say, don't call it research, we'll call it tomorrow's treatment. And I look at this and listen to what Eliot said, and it sounds very futuristic. We get for this down there was no transplant before we did it. There was no bionic heart before Daniel did it. You're going to see this move forward, and the fact that the Wesley stand is impact. Eliot and Arutha puts his head with the rest of Australia. I just tell the story. When Eliot came across in UPIT, he was going to Sydney and Melbourne, and then he kept hearing about Brisbane. And there used to always be people who went to Sydney, Melbourne, Sydney, Melbourne, Sydney, Melbourne. But we managed to attract them up here, and Australia, the Queensland, Brisbane, is leading and is the MP. So as a chubby, middle-aged Glaswegian, I have one moderate, self-interest, cardiovascular disease. Normally we die when we're about 51. We don't, the Queen doesn't send us a telegram or 100, we get up for 50. Complete self-interest. But I think, when Dave started doing transplant, Daniel started doing bionic hearts, all these things, people saw these as sparrows, never nevers. This isn't never never. And the facts that the others in News and the Wesley, CCRG, IAP, we're getting the brightest people. It's a tsunami of disease that's just getting bigger. And what'd be lovely is 10 years' time, that man doesn't present TBD. I'm not up and see in the morning, and I look as good as Andrew Parr.
Just speaking about the work that happened during a pandemic, we went in thinking COVID and the flu wouldn't be the same in the way it affects the heart. And what we found, so we thought inflammation is going to be through the roof. We got these tissues brought over from Brazil and multiple places around the world. They were run in the US because we didn't have access to the platforms in Australia. And we went in blind. Inflammation is going to be through the roof in these patients that have died. Or what we found in the cardiac tissues was that DNA damage was up, but the inflammation was suppressed in these cardiac tissues. So it was almost like flu, severe flu, and COVID was impacting the heart differently. And this was only possible because of enabling this technology to almost unleash this technology on this disease. And so what was really interesting was we could start to understand the molecular fingerprint that was underpinning a lot of this.
And just to highlight the fact that make the All Blacks feel too guilty in the front row, I can understand this. These two are the clever blocks here. So we were sending this tissue away to America, and I said, why are we not doing it here? So we do have one of the machines. Why don't we have one of the machines? We don't have any money. Okay, we'll go get money. And credit to the generosity of the All Blacks, the Fitzgerald family, St Andrews, and the Wesley. That's how we did the first machine in the first hospital in Australia. So never waste a good crisis. And if you want to get money out of a stone, you get a Scottish guy, and then you get two really clever people. So Lyndon, I'm coming for you next. But at least have this machinery anywhere in Australia. And that's where, looking at this tissue, realizing flu doesn't affect the heart the same as COVID. Flu causes inflammation that kind of burns out like a burn on a farm. COVID changes the DNA. There's all these things we don't understand. Without the ruler, the measuring stick, we can't do it. Now we're at the forefront of Australia, and with Luther's professorship, the forefront of the world. I have brought Eliot here. We're in a fantastic position, not just for the Olympics, but to win something much more important, that's the advancement of cardiovascular disease.
So we might go to why we're here today, Eliot. Eliot, what made Brisbane attractive to you? You went around Australia, you visited multiple sites. What was unique about Brisbane, and maybe speak about third room?
Yeah, happy to. I love Brisbane. It was not an accent. It was not an accent. Go Rose.
No, what happened was you get connected. So when you live overseas and you hear about Australia, your understanding may be limited at first, and your connection points may be in the two big cities further south here. And I visited those places, and they were interesting. And I remember when I came over with my family to visit the major research institutes at those places and give talks and get offers and things like that, we decided that we should also make sure to stop by Brisbane on our trip here, mostly because it looked lovely in pictures, but also because we were traveling a long way. This was a big decision. We wanted to make it thoughtfully. And I remember I didn't have the original points of contact here that I had in the other cities. So instead, I just sort of got on the internet and on this guy, and I emailed him. That's what happened. And I said, we're planning a trip over to Australia for some job interviews and to just explore job opportunities there. Do you know of what might be going on at research in Brisbane? As a matter of fact, I do. Just about nothing here happens without him knowing about it. And so we made sure to add a Brisbane journey on our trip over here. I came up here, and a couple of things impressed me immediately. The first thing that impressed me was after visiting Melbourne and Sydney, I had attempted to bring up the idea of AI and spatial biology at the cardiovascular institutes in those cities, and blank stares. They were utterly unaware of the technology as it could apply to their diseases. And when I came up here and talked to people, they said, we've been thinking about this for a while. We are the leaders in terms of having the most instruments, the most expertise, the most publications, and the most interest, not just in cardiovascular spatial biology, but spatial biology in general in this country. It's obviously that was very impressive to me. The other thing I immediately noticed was one of the first things that John said to me when I visited with him, and it was supported when I first met Andrew and everyone else, was that we're trying to do things differently in Brisbane. Down in Sydney and down in Melbourne, I got the distinct impression that if you were at one institution, you were within that institution, and that is your home, and you are in competition with everyone else in the city. But that was not the message here. The message here was we are trying to build something bigger. We may be a little smaller than them, but if we all pull together, we can make a bigger impact than any of them. And that was the vision that was sold to me, and it's fully lived up to it. The partnerships between private research institute, between universities, between public health system, people are actually eager to do that here, and it's making a difference. And it's going to pave the way for this city to lead research in the next decade. I am certain of it. And it makes for a much more exciting environment. I couldn't be happier.
We might just see if anyone's got a question, the audience.
There are two messages. One is personalized medicine, personalized heart forecasting, and the other is the problem with cardiovascular disease is that it really begins before the symptoms occur. Addressing the second of those things, how do you see the future? Many of us go to the doctor periodically, having done blood tests and whatever to see what our general health is like. But how are we to know that we have embryonic heart condition ahead of the symptoms? How are we to know before our arteries harden? How are we to know before the symptoms occur? I heard nothing tonight that gives me confidence that when I go to the GP next month, I'll be able to say, hey, have you found anything that indicates that I've got preliminary indications of heart disease? And that's one of the two messages that you are putting out this evening.
Can I also? the societal thing.
Sure. See if I answer it here, and if not, I will clarify further. So I think those are the two of the core messages to take from this, right? And they're related messages. Personalizing, to personalize someone's care, you need to know where they're at. And so knowing where they're at, our goal is to, yes, personalize care, but part of knowing where they're at is to be able to identify things sooner. Certainly for certain stories that I told tonight that I think comes across in the case of transplant where we're getting their tissue regularly, we really are forecasting months in advance, sometimes years in advance. So we are getting a window into their future. For other diseases like coronary disease, I'm not going to recommend that we go around and take tissue from every 30-year-old and every, from everyone's hearts in the world to figure out whether they have the early signs of heart disease. That's not a practical solution for that problem. But what we learn from developing unique experiments like the one we conducted where we access unique tissue cohorts of people who have early stage disease is we learn about the biology that's present early on. We can already give people somewhat risk, we could argue some risk prediction as to whether you're at risk for coronary disease in 10 years. Our profiles are okay for that, but we look at your cholesterol and your family history and your blood pressure. And so we can say you're a high risk person or a lower risk person. We already do that. But it'd be great if we could do something other than put you on aspirin and a cholesterol lowering drug. If we actually had something that targets the biology that's not working properly in the heart itself, instead of dealing with just the systemic problems, high blood pressure, high cholesterol, that would be a major breakthrough. Currently, none of our coronary artery disease drugs are focused on the heart muscle cells themselves because we don't understand the biology there. So in order to make that kind of breakthrough for the type of disease maybe you're referring to, our studies are slightly different. They focus, we're not going to biopsy your tissue, but we are going to build unique experiments that teach us about the biology that will matter for people so we can start them on treatments sooner, prevent disease from reaching that critical stage where they're symptomatic. And I think maybe that's the way I'd sort of envision it mattering for your GP.
I've been 5 children. So the thing that I used to get asked a lot in the back of the car was, are we there yet? We're not there yet, but we're getting there. So I think of when I qualified to try and get a cardiac ultrasound, you get one in 2 or 3 hospitals in the country. You get angiograms, a jab in your groin, a catheter up there, and you could maybe get ECG. Today, you can get a CT coronary angiogram, no jab down there. We had a patient literally at 5 o'clock who came in with chest pain, it was unclear. We'd get an immediate blood test. Not predicting what's going to happen in 6 years' time, but it is telling us within 30 minutes we got a highly specific troponin. We can get a cardiac ultrasound to show that the heart muscle has not been damaged. There's biomarkers in our blood tests, we naturally peptides, etc. So medicine is not it's not like television. Research is not television. It's not razzle dazzle. It's sequential small steps, and the more steps you put together, and I have to be careful about many here, it's about building bridges, not walls. Sorry, mate. It's about building bridges, not walls. But if we all put a brick in the bridge, then the stuff I talked about that didn't exist 10 years ago, you come back in 5 years' time, and we'll be ahead, and we'll be ahead. And that is it. You're right, we're not there yet, but the stuff we can do today, we could do 5-10 years ago. When we give the transplants, that was the never. So it's an ongoing journey. It's not like a 100-meter sprint and we're finished. We will just keep building. We stand on the shoulders of giants, and that's what philanthropy allows us to do. I think also just the beauty. Just think talking about the ultrasounds, ECGs. Imagine creating a digital patient, bringing your echocardiogram, your ECG, your biochemistry, some blood tests, and bringing it together. Because AI is just massive amounts of statistics. If we bring all these data together and create a digital patient of you where it combines and each part of it has got a predictive value, then we get there much quicker. And you maybe want to talk about that. Thank you, John.
Part of the work we're doing as well is we're collecting bloods and tissues from patients as well, and we're collecting it earlier and earlier on. And so when we work with the biobank, we're biobank healthy samples, we're biobank disease samples, and there is a point at which you go into disease and then patients stay healthy. And so it's ability to do all the cutting edge tissue mapping, identify your biomarkers, but then also derive that from the blood. And so we do a lot of blood studies. We published a big pulse read looking at 8,000 individual proteins in the blood or 10 microliters of plasma. So we're continuing the blood work in parallel to the tissue work as well, so we can discover new biomarkers, but ultimately the tissue biomarkers will dictate what we look for in the blood. So I hope we've answered your question. We might pass to...
Right, there's one thing that I just like to add to this, and I know that maybe I'm going outside the square, the brief. Okay, so I'm sorry about that. Have you looked specifically at genetics? Because I'll just use us as an example. My husband's genetics have been passed from his father to him and to our son now, who has problems with his heart, but blockages. It seems to be genetic. I have a lot of Scottish genes. But unfortunately, Scottish genes seem to include getting mitral valve prolapse with regurgitation, for which I had 2 operations and I've got further problems. So do you look at people's genetic background? When Colin's asking the question of how do I know, but is it important to look at what your father died of, what your grandfather died of? Are you Scottish? Because mitral valve prolapse with regurgitation is a very much a Scottish thing. I'm sorry. But if you can answer that in any way to extend on what Colin was asking.
So we'll start by saying that Lord Brisbane came from Glasgow. We're not the First Nations people, but we're the Second Nations people. I think this is terrible. Yeah, there are peoples though. There's a great paper by a chap called Sorensen in 1988, going back a bit, and he looked at identical twins in Norway, but they've got a fantastic record. And again, this is the power of huge data. You've got a database. So they looked at identical twins, perfect for genetic association. And you're right, we always think, like Scottish chips, like deep fried Mars bar, and did your mum have a heart attack? And we always think that there's a very strong genetic association. He showed that between the twins that were separated at birth, some went to a rich house, some went to a poor house, your risk of cancer, looking at genetics, was about 1.05, an association. But if one twin died before 40, there was an association, but it wasn't big. Cardiovascular, it wasn't that big, which flew in the face of everything. The biggest association genetically was your response to infection, and this is an area. So genetics is definitely a component, but what's happening at the tissue level and what this technology allows us to see is beyond genetics. Genetics determine a part of this, but as Eliot said, we should think the sarcoid story. We misdiagnose it, we can see genes, no problem. But the genes in your hair, if I had some, the genes in his hair, are the same genes that are in the skin, but skin doesn't behave the same as hair. So there's a whole pile of things that happen. The transcriptome, which has got transcriptomics, comes from. So it's imagine looking without their glasses, a small thing in the screen, or having a microscope and being right. So yes, genetics is a component. No, it's not as big a component as we thought. You're very, very lucky not to be English.
Just upset one of the audience.
That's a great answer. And the answer I give, whether it's to a patient or just someone asking in a seminar like this, is of course your genes matter. We've known they matter for a long time, but they're not the whole story. And we've known that for a long time too. I was telling people last week, I told patients many times, your genes are the hand you're dealt, and then there's how you play it. And they both have a big impact on your health. There are many people who have a risk for high cholesterol, and then there are many people who make that risk manifest in early heart disease by smoking cigarettes, non-exercising. and a bunch of other choices, living in poor socioeconomic conditions and many other things. Genetics is a piece. They're all pieces of a puzzle. Biology is a complex story. Genes are a foundational piece, but genes assemble themselves. Genes make transcripts, and transcripts make proteins, and proteins build themselves into little structures we see in a microscope. And then that little structure forms organs, and organs form people. And so you want to understand all of that if you want to properly predict things and understand things. So I want to understand the patient level experience, their environment, how they eat, how they live. I want to understand their genes. I want to understand their transcripts, their tissue biology, their tissue proteomics, and everything in between. Because you skip steps, you miss signals. And we're trying to fill in a key missing step with these tools. And in fact, it might be the biggest step since the discovery of the genome.
Any more questions from the audience? Oh no, Russell? I see Russell so putting up his hand.
That is can add up. So, perhaps just a general question is the time we expect some sort of development to come through, but worthy good double the chances are it has been another it's essential thing what a quick outcome there is on society. Because I might be clinical quite soon. Is there a time frame that you're looking at with your work? The one in Australia or sign off?
Yes, there are timeframes. And depending on the project, the time frame is of course different. So for the sarcoidosis story I told, we already have program currently run on the machine upstairs on the 8th floor at WRI, where we're trying to take that initial discovery, reduce it down to a smaller panel of markers that we can actually run on clinical tissue. So the translational arc for that project, for that program, is short. I'd like to have a potential candidate clinical diagnostic within 18 months. That's my timeline for that. And then you have to go through the process of establishing it in a lab or in a clinical lab and making it a real diagnostic. But that is a short timeline project. Transplant projects probably a little bit longer, but we've come a long way over the years, and we're targeting increasing implementation exercises where we're trying to figure out how to get all of this captured data from patient level to the G level, to the tissue level, to the ECG and the echo, put it all up in the cloud, use our AI tools to integrate it and get individualized predictions. We've done it in a research database. It seems effective, but doing it in real world clinical workflows requires that next step. We are working towards those steps. So that timeline a little longer, but still not a long, hard horizon. When it comes to identifying new drug targets for coronary artery disease, we may have found some pretty cool mechanisms, but the walk from a cool mechanism to a drug that I put in a patient is a longer arc. And so it's very project specific, but for sure as a clinician, I don't embark on a project that I don't see a translational arc on because I get bored quickly.
Diagnosed EDD, right? What you diagnosed, thanks, mate. When we started this project, I showed some maps up there. This was how could we get a heart to be transplantable for longer. You've got about 4.5 hours to get it from the donor to the recipient. And these people, the donors of the organs, are the most brilliant people in medicine that allow us to do everything. But Australia's a big country. So getting a heart from that young lady, I showed the picture, she was Perth, Western Australia, David went and saw her. And that heart had to stay outside the body for almost 9 hours. That wasn't doable until some of the work we did in the animal lab for 4 or 5 years. And then with David McGiffin, colleagues at Alfred, we changed the way the transplant happened over 4 years. So almost every heart now in Australia is transplanted off that box that we worked on for 4 years. I'm the same as Eliot, that's not going to affect a patient. And I agree the transplant program is maybe a bit further away, but just yesterday, day before, we need lots of tissue samples. and talking about Queensland is a place that works well. There's about 10 of us on the phone now. I think there is a Queensland tissue bank that have you using the cardiac tissue. So yesterday, with the heads of the tissue bank, with Eliot, myself and Arnold, the other scientific team, we now have a way to access data that donors have been giving to the tissue bank for years. And just to finish, we had a difficult patient over Prince Charles transplant. The heart, the donor, was put on the rig that we worked on for 4 years at CCRG, was transplanted at Prince Charles, worked beautifully for the first 10 hours, and then it just started to descend. And the patient looked like they were going to die. Great work at the Prince Charles teams. They've survived, but we have no idea with our normal biopsies why that heart went south. So we said to the team, we don't know that the techniques we've got today don't tell us. What we do have now is the samples from that patient that they've given care, that's sitting in our cells also. We know, but we took those samples that are being taken and we're analysing already here. And we've got normal tissue for patients that get transplant, and we're now comparing and contrasting. There's only one patient, but if that's your son or your daughter, that's a really important question. And 2 years ago, or a year ago before we got it here, we wouldn't have an idea, and that person's living with a transplant, that he or she doesn't know whether it's going to stop in a year. But we're going to be able to tell that stuff from this technology. So it's one patient. It's not a cool journey, but as Mandela says, a journey starts with one step.
One last question. John, I'm enjoying this because I can ask John and Eliot questions that, they're forced to answer. John, you've by a core, to artificial heart. You've gone through this journey before. What is it about spatial medicine that you think is going to potentially define the next 10 years of biomedical research? Do you think it's that? And do you see, is this a convergence of science, AI, tech, and medicine? Do you see that playing out in close ever? Sure.
So I really am the dummy in the spatial medicine. These two are the clever clerks. I'm the dummy. But I remember Aretha would speak quickly and I wouldn't understand it. And I hate being the dumbest person in the room. So I went on to Spotify to listen to a whole pile of how it got there because he's too clever. And it was about how the cloud computing actually allowed this field to develop, because the amount of data that's involved in one of these slits are terabytes of data, and you can't have Andrew Wood bias a computer that's big enough for this. So you're using cloud data. So without a cloud, this technology wouldn't happen. So it's not just a microscope and a clever scientist or a medic or two. It's computing. It's using massive amounts of data. It's bringing clinical, radiological, biochemical, and rather than trying to make a spaghetti bolognese with just meat, this brings all the aspects together, the mince, the tomatoes, the carrots, the pasta, brings it all together, because a patient isn't one thing. We're all amalgamation of all part of it. So if we can bring all these data streams together, rather than in silos, the radiologist says, oh, your ultrasound looks fine, the ECG person says your ECG looks fine. Imagine we can start talking with it all together and create a digital patient. So that's what I think computing, massive amounts of data, collaboration, and with Queensland, Brisbane, QSBC, Wesley Research leading Australia in the world.
I completely agree with what John said. This is the reason I think that the technology of the next decade is because it's tight. The world is ready for it. The computational infrastructure is there. Everything in our lives is already digitized. That was happening anyway. Now we have the tools to bring it all together and to mine it for hidden information that matters to patients. The moment is right. It's like Eddie, fortune favors the prepared, and by embracing these technologies early and recognizing the moment, we can harness this and lead its deployment in real patients' lives. And that moment is here. I think it is inevitable, and I think we have a team that can lead it.
We might close the panel and fast it over. So thank you.